Hello everyone. So we're actually going to film the lab we're doing in class today, and we're setting it up right now. Miss Manzano, if you want to say hi. Hi. These are just cups of water, and in each cup we're going to put a piece of dialysis tubing. So this is the semi-permeable membrane we talked about yesterday when we had our virtual class. These have to soak in water before you can open them and use them. So we're just going to soak them before the students come in. Okay, so here's my starch and water. I'm gonna put it into the model cell. Remember, this is dialysis tubing. It's plastic that has pores in it, so it's going to be semi-permeable. Now I'm just adding some sugar water, and I'm gonna put it in so that it's low enough down that it doesn't leak out. Again, if you're trying this at home, nothing here is harmful. It just might spill, so maybe try it over the sink. Okay, so now this is what it looks like. I have, the bottom is sealed because I folded it up and tied a knot. The top, I'm gonna do the same thing. So I'm gonna fold it down, and this part, you kind of have to be nimble with your fingers. So you might need somebody who's around to help you hold it, or you can try to give it a good seal, lay it on a surface. It might leak a little, but that's okay. So if you need the tutorial for how to tie the bottom, here's how you do it. So it's folded over. I'm gonna take the string, kind of wish I had a third arm right now, that'd be helpful. And I'm just gonna loop it around there, try to make sure I get a good seal. I can lay it down, it won't leak out too much because we're gonna rinse it off anyway. So if it's easier to see from laying down, I'm just gonna pull the string under it a little bit. Again, hard to hold my phone and do this at the same time, but I will tie this part closed, and I'll try not to get too much liquid in it. Now I tied it closed, and I had to lay it down because I don't have a third arm to tie it closed. And so when I did that, some of the starch and sugar liquid leaked into the top. So I'm just gonna rinse it off. This is fresh water. You could rinse it under the sink if you have that. We just don't want to cross-contaminate our substances because we won't know if the lab worked. So both ends are sealed, tied. Inside we have starch and glucose. Okay, now I'm gonna drop that cell that I made, here it is, into just a beaker of fresh water. This is supposed to be a clean beaker of water, and I'm going to add my iodine. So we wanna add enough to turn the outside water yellow. So if your iodine is kind of faint, you might need more, but as long as it's kind of light yellow, a few good squeezes of the dropper bottle from your kit, and we're gonna watch and wait.
15 minutes later.
Okay, so we've watched and waited, and now I'm doing this with a travel group that's actually here, but they're taking a break outside. Here's what some of them look like about 15 minutes later. So if you're watching and waiting for yours to change, okay, so yours might be light purple by now, or if you left it for a while, it's going to be black. Here's a setup. Oh, there it is, floating in the cup. Again, iodine is not harmful. It's like Purell, so you can reach in, or if you want to use a spoon or the pipette to pull it out, you can. Here's what it looks like now, okay? So it's turned kind of purple. If you put in more starch and let it sit longer, it might be black. Here's one that looks a little bit darker, and we can take it out of the cup, and you'll see that it's a little bit more closer to black than it is to purple. Here's that one, but there's definitely a change that happened. There it is in the cup. So next, we want to know if the outside liquid has anything besides iodine in it. So we can see from the cell that the iodine went in, it turned the starch black, and we can see from the outside there's definitely no starch because the outside would be cloudy black or purple as well. But we can't see glucose, and we're gonna have to test that with a different indicator because iodine only tests starch. So let's try that. So as a refresher, this blue liquid is Benedict's, and I just mixed it with some water because this is what a negative test looks like. So Benedict's does need heat for it to react, and we won't know if this has sugar in it until we see, but just so that you can see a control, remember the original, what it just looks like. Benedict's and water, a negative test. If there's no sugar in the outside liquid, it's going to stay this color blue when we heat it up. And I want to test the outside liquid. So I'm going to fill this test tube with solution from the outside of the cell. And I'm just adding it to a clean test tube, which I'll show you in a moment. So right now it's yellow because of the iodine. There it is. And I'm going to now add some blue Benedict solution. That's the test for sugar. And remember, Benedict's has to work with heat. It won't change. So even if there is glucose in it right now, it's not an instant reaction like iodine would be. So we're gonna put it into this water bath. This is how we can heat a test tube safely. When this is not that hot, I just put it on, but when it's boiling, we'll look for a color change. So we'll come back to that in a moment. Okay, so here's that test tube we put in the hot water bath, and It's kind of see-through, transparent, which is not like the previous tests we've done, but you can tell that this is definitely in the red-orange family because that's a red piece of paper. Pretty similar. All right, I'd say that's a positive glucose test.